Well, welcome to the first lecture on protein structure. I believe now you are conversant with amino acids because some of the concepts that we have talked in the lecture on amino acids will be required for you to understand protein structure. What we are going to do in this lecture is to look at peptide bond in general. Then we will look at two levels of protein structure, the primary structure and the secondary structure. And towards the end of the lecture, we will look at supersecondary structure and some associated definitions. Now, before I go into the lecture, I would like to highlight that in this particular lecture, you will require to refer to your textbook in biochemistry. That is Lippincott. A soft version in PDF format of this textbook is already uploaded on the LMS. Of course, the uploaded version is the 5th edition of Lippincott, but I believe it will serve our purpose. And when you are reading these particular sections, which I will highlight while I go through this lecture, I will urge you to take notes. Now, before I talk about the peptide bond, let me just quickly revisit a very basic concept in organic chemistry, that is the functional groups. I believe that you have already covered this part with Professor Bayumi. So we have looked at carboxylic acid group when we have talked about amino acid. We have also looked at the amino group when we have talked about amino acids. You may have already looked into aldehyde and ketone when Professor Bayumi would have talked about carbohydrates. For example, glucose is a carbohydrate molecule that has an aldehyde group, whereas fructose is a carbohydrate molecule that has a ketone group. When I take a carboxylic group and an amino group, and when these two groups react, we get the amide group. And this amide group is what is involved in the formation of the peptide bond. That means the reaction of a carboxylic acid group with an amino group leads to the formation of an amide, and this amide is what is the basic structural and functional composition of the peptide bond. Let us look at this. How is an amide formed? So I take a carboxylic acid group, I take an amine group, and when I have proper reaction conditions, we are not going to look into the detailed chemistry, but when we have the ideal reaction conditions, the carboxyl group reacts with the amine group. There is a loss of water here, and this leads to the formation of an amide. Let us now translate this concept between two amino acids. Sorry for this kind of crowded slide. So we take two amino acids. This is amino acid 1 and this is amino acid 2. This is the carboxyl group in amino acid 1. This is the amine group in amino acid 2. And the R1 and R2 are the side chains, which you already now know. And when I have a reaction between the carboxyl group and the amine group, there is a loss of water, and this leads to the formation of an amide. This amide linkage between two amino acids is defined as a peptide bond. And what I get here is a dipeptide. Di meaning two. So in order to form a peptide bond, I will always require 2 amino acids. So the concept here is we can never have a monopeptide, because you cannot have a peptide bond formation with only one amino acid. Similarly, let us take a hypothetical peptide which has 3 amino acids. So how many peptide bonds will be formed between 3 amino acids? The answer is 2. So in a tripeptide, that means when I have 3 amino acids, tri is equal to 3, I will have 2 peptide bonds. Again, I come to the concept that I mentioned in the previous slide, that the formation of such a linkage requires the removal of water, and therefore formation of a peptide bond requires dehydration. Dehydration, loss of water. Now, the most important concept associated with the peptide bond that you should always remember is that a peptide bond is a covalent chemical bond. And since it is a covalent chemical bond, it is a very, very strong bond. Now let us look at this tetrapeptide. Why tetra? Because you have 4 amino acids: alanine, glutamic acid, glycine, and lysine. So what is happening here is we get a peptide bond between the carboxyl group of alanine and the amino group of glutamic acid. That's peptide bond number 1. Then when I come to glutamic acid, the carboxyl group of glutamic acid reacts with the amino group or the amine of glycine, and I get the second peptide bond. Now let us come to the third peptide bond, where the carboxyl group of glycine reacts with the amino group of lysine, and we get the third peptide bond. So at any point of time, you can see that one amino group and one carboxyl group in a peptide is free, and this can also be translated to proteins. So proteins will always have one free amino group and one free carboxyl group, and this amino group and this carboxyl group will always be at opposite ends of the chain. So if I have to read the sequence of a peptide, I will always start from the amino end and go towards the carboxyl end. Sorry. So how will I read the sequence of this tetrapeptide? The first amino acid is alanine. The second one is glutamic acid. The third one is glycine. And the fourth one is lysine. So this particular peptide has 4 amino acids. So it is a tetrapeptide, 4 meaning tetra. And, sorry again, there are 3 peptide bonds. This is the first one, this is the second, and this is the third. So if I have a peptide with 5 amino acids, that is a pentapeptide, I will have 5 minus 1 is equal to 4 peptide bonds. So the number of peptide bonds in a protein or a peptide will be equal to the total number of amino acids minus 1. So if somebody asks you how many peptide bonds are present in a peptide containing 10 amino acids, the answer is 10 minus 1 is equal to 9. And each peptide or a protein will have one free amino terminal and one free carboxy terminal. And if somebody asks you the sequence of the peptide, you should read from the amino terminal and go towards the carboxyl terminal. And this is very important because we generally ask questions on sequence of a particular peptide in an assessment, and there you require to read the sequence of the peptide. Now, we talked about this in our class, that when you have a peptide bond, the carboxyl group and the amino group are trapped in an amide bond formation. So the only free part in an amino acid is the side chain. So the only free part in an amino acid when it is in a peptide or a protein is the side chain. So the side chain of amino acids in a peptide or a protein contribute to the overall acid-base properties of the molecule. That is the reason we always focus on the side chain of amino acids, because amino acids, mostly in the physiological environment, remain as part of a peptide or a protein. So the carboxyl group and the amine group are not free. What is free is the side chain. And I think this concept should be clear by now. Before we progress, I just want to highlight one aspect. And this is, what is the difference between a peptide and a protein? A peptide, generally, conventionally, people say, consists of lesser number of amino acids. And the convention is, if the number of amino acids is less than or equal to 50, we call it a peptide. So less than or equal to 50 amino acids, you get a peptide. And if something is more than 50 amino acids long, you get a protein. But the bond or the type of bond in proteins and peptides between two amino acids is the same. That means there is an amide linkage between the carboxyl group and an amine group between two amino acids, and this leads to the formation of the peptide bond. And I believe this concept should be very, very clear, because without this concept, you will not be able to understand the different levels of protein structure. And the other concept that you should always bear in mind is that the peptide bond is a covalent chemical bond. So what are the key features of the peptide bond, and that you must always remember? First of all, I mentioned this, peptide bond is a covalent bond. That means it is a very strong bond. And it has a partial double bond character. You know what is a double bond, what is a triple bond. So it has a partial double bond character. Secondly, since it is a very strong bond, because it's a covalent bond, You cannot break a peptide bond very easily. You cannot break a peptide bond using conventional protein denaturing agents. And what is protein denaturation, we will talk about in the next lecture. But what we do when we denature a protein, we apply heat or we treat the protein with high concentration of salts. If we apply heat to a certain extent, and when we treat the protein with high concentration of salts, we will not be able to break the peptide bond. But how can we break the peptide bond? You can do so by exposing the protein to very strong acids or bases. When I say strong acids, such as hydrochloric acid, nitric acid, which are very strong acids. When I talk about bases, sodium hydroxide or potassium hydroxide, which are very strong bases, or by exposing the protein to very high temperature. When I talk about very high temperature, I mean high heat that will completely denature the protein as well as break all bonds, even the bonds in the amino acids. So what you will get at the end are atoms like carbon, hydrogen, oxygen, sulfur. Or you can treat the protein with certain biological catalysts which are known as enzymes, and such enzymes are involved in the digestion of proteins in our body. This particular part, how enzymes are responsible for breaking peptide bonds, you will cover in detail in the digestive system. So coming back, peptide bond is a very strong bond which is resistant to high concentrations of salt and moderate heat and requires very harsh conditions if you want to break that bond. Now, what I want you to do is to take your textbook and in the textbook read characteristics of the peptide bond. And one thing that I will like you to focus on is that the peptide bond is generally a trans bond. I believe you have already covered cis-trans isomerism, and a trans bond is always more stable than the cis bond. And that is because there is a phenomena called steric hindrance or steric interference happening between the side chains in the cis position. We will talk about this steric hindrance and cis-trans isomerism in bit more detail in our in-class session. But what I want you to do for now is to read this part in your textbook and make some notes. Now, let us try to solve this question. This is like a self-assessment. Which of the following statements best describes the difference between a polypeptide and a protein? We have four options. So polypeptides consist of 100 amino acids, whereas proteins consist of less than 100 amino acids. When I talked about the difference between peptide and proteins, I told you that proteins contain 50 or more amino acids. So this part of the statement directly tells you that this is not the correct option. Polypeptides do not exhibit physiological function, whereas proteins perform designated physiological function. Proteins perform designated physiological function. And what are polypeptides? They are multiple amino acids joined together by peptide bond. And peptides also can have physiological function. We talked about briefly about antimicrobial peptide in our lectures on amino acid. So polypeptides do exhibit physiological function. That means peptides do exhibit physiological function. So this particular option is also wrong. We come to the fourth option. All polypeptides are sensitive to pH and temperature. We talked about side chains of amino acids being responsible for their ionization and sensitivity to pH. Polypeptides also have amino acid side chains free, so this particular option is also wrong. So the only remaining option, all proteins are polypeptides, but not all polypeptides are proteins. This is a typographical error, so we just need to correct that. So all proteins are polypeptides, but not all polypeptides are proteins. And that's the correct option, because when we talk about proteins, they are actually having multiple peptide bonds. But polypeptides also have multiple peptide bonds, but they may not have the required number of amino acid that you need to form a protein. So this is kind of a take-home statement. All proteins are polypeptides, but not all polypeptides are proteins. Please remove the not here. That's a typographical error. So the statement should read, all proteins are polypeptides, but not all polypeptides are proteins. So we have looked at the formation of a peptide bond. We have looked at how to read a peptide sequence, how to read a protein sequence. We have talked about how we know how many peptide bonds are present, the number of peptide bonds present in a protein, by knowing the total number of amino acids present in the protein. So now we go to the different levels of protein structure. The first one is primary structure. How do you define a primary structure? Primary structure simply refers to the linear sequence of amino acids joined to each other through peptide bonds. So if somebody asks you, what is the primary structure of this polypeptide? So what you need to do is to start reading the amino acid sequence from the amino end. I mentioned this previously. You should always read the amino acid sequence from the amino end. So glycine, proline, threonine, glycine, threonine, glycine, so forth and so on. So the sequence of amino acids determines the basic structure of the protein. This is very, very important. What is primary structure? In one line, it is the sequence of amino acid in the protein. What is the bond that is involved in the formation of primary structure? The answer is peptide bond. Please remember this, because in assessments this is often asked. So primary structure is equal to the sequence of amino acids. The sequence should be read from the amino end towards the carboxyl end, and the bond that is involved in the formation of primary structure is the peptide bond. No other bonding is to be mentioned when somebody asks you what is the bond that is responsible for the formation of primary structure. So we move on, and this year we need to talk a bit about variation in primary structure, because this concept is very important, and this you will see in the tutorial session why this concept is important. Now, within the human species, the amino acid sequence of a normal functional protein, that is, the primary structure of a protein, can vary even among individuals, even among the tissues of the same individual, even at the stage of development. So you may encounter minor changes in the primary structure of the protein, and these changes can be classified into three: variation among individuals, variation in the primary structure of the protein in the tissues of the same individual, and variation in the primary structure of the protein at different stages of development. Now the question comes, okay, I am observing variation, but are these variations tolerated? Because if you change the primary structure of the protein, the function of the protein may also change. Now, in a protein, there are certain regions called non-critical regions. That means if I change the amino acid sequence in these regions, the function of the protein is not altered. And the change is tolerated if the substitution is a conservative substitution. I believe we have done a mock exercise on this. And what is a conservative substitution? When we replace one amino acid with another one of similar structure or chemistry. We talked about this using the example of isoleucine and leucine. So if I have a conservative substitution, then this particular change or variation in the primary structure of a functional protein is well tolerated. Let us take an example. This is the three-dimensional structure of a protein called factor 10A. Factor 10A is a protein that participates in blood clotting, and I mentioned blood clotting, why it is important. Now, these are different regions of the protein. Now, let us say this particular region of the protein is called the GLA domain. The GLA domain, or the gamma carboxyl acidic domain. We talked about what is gamma carboxylation. So this particular region or domain of factor 10A has gamma carboxyl residues. If you change the sequence of amino acid in this particular region, so let us say you replace alanine with glycine in this particular region, the function of factor 10A is not affected. So that's the reason I have put a green bracket here. So this is a region which is a non-critical region. But if I change the primary structure in this particular region, the function of the protein will be drastically affected. So this region is a critical region. So any amino acid change or any change in primary structure in this particular region will be tolerated, whereas any particular amino acid change in this particular region will not be tolerated. So, depending on where you change the primary structure, a particular change in amino acid sequence will be tolerated. If I change it in this particular region of factor 10A, it will be well tolerated because this is a non-critical region. However, if I change the primary structure here, it will not be tolerated because this is a critical region. So depending on where you change the primary structure, a particular amino acid change will be tolerated. If I change it in the non-critical region, then it will be well tolerated, whereas if I change it in the critical region, it won't be tolerated. Now we go to the concept of substituting an amino acid with another one. So let us take a peptide, glycine, isoleucine, valine, glutamic acid, and glutamine. And this is the amino terminal end. This is the carboxyl terminal end. So I am reading the sequence from the amino terminal end to the carboxyl terminal end. So now I want to make a substitution at the position of isoleucine. So what should I do? Isoleucine, by now you already know that it's a hydrophobic amino acid. I will change this particular amino acid with another hydrophobic amino acid such as leucine. So this is a conservative substitution and therefore this will be tolerated. Next, I change isoleucine to valine. Valine is also a hydrophobic amino acid, not as hydrophobic as isoleucine, but still hydrophobic. Therefore, this particular substitution will also be tolerated. However, next I change isoleucine with lysine. Isoleucine is a non-polar amino acid. I have changed it with lysine, which is a polar amino acid. So I have completely reversed the property of isoleucine from non-polar to polar lysine. As a result, this particular substitution won't be tolerated. So if I am doing a conservative substitution, a change in the primary structure will be tolerated. But if I am not pursuing a conservative substitution, or if the substitution is non-conservative, a change in the primary structure won't be tolerated. So this concept should be clear because in the exam we may ask you a question that if I want to have a conservative substitution in this particular peptide where I want to make a change at the position of isoleucine, which of the following amino acids will you require? And you are given a choice, and what you need to look at is another amino acid which is non-polar. Now, the primary structure of the protein is determined by the genetic code, and this brings us to the concept of central dogma. I'm not going to cover central dogma in detail in this particular lecture because I believe you will revisit the concepts of central dogma again in molecular biology and principles of genetics. So what happens is you have the DNA, the sequence of the DNA determines the sequence of the protein. So the central dogma tells you that based on the sequence of the DNA, the sequence of the protein or the primary structure is determined. If I want to go through the steps, DNA undergoes a process called transcription, which requires the enzyme RNA polymerase. This leads to the formation of messenger RNA. Then messenger RNA undergoes a certain set of chemical reactions called splicing, which is mediated by a multi-subunit entity called the spliceosome. And this mature mRNA then undergoes translation, and in the process of translation you require transfer RNA and ribosomal RNA or rRNA and the ribosome, and translation leads to the formation of protein from mRNA. So DNA, transcription mRNA, translation protein. The sequence of bases in the DNA determines the sequence of amino acids in the protein. That means, what is the determinant of primary structure? DNA is the determinant of primary structure. Now, I'm not going to focus, as I said, on individual processes of transcription, translation. These are part of molecular biology, but what determines the sequence of amino acids in a protein is the genetic code or the DNA. Now, after primary structure, we come to the second level, which is called secondary structure. So primary and secondary. So you have a sequence of amino acids. You have a thread. This thread consists of multiple amino acids, and you are calling this thread a protein. But this thread does not exist as a thread because segments of this thread will start folding. And what is secondary structure? Secondary structure is defined as the folding of 3 to 30 amino acids into continuous segments of polypeptides into geometrically ordered units. So secondary structure is defined as the folding of short continuous segments of polypeptides into geometrically ordered units. Now there are two types, alpha helix and beta pleated sheet. And you can see here helix has a helical structure and beta pleated sheet has a sheet-like structure. If I want to understand secondary structure in a bit of detail, so here is the sequence of amino acids. That's the primary structure, and this is the single letter code for amino acids. So methionine, leucine, methionine, leucine, isoleucine, leucine, so forth and so on. And as you can see here, this particular segment will fold 3 to 30 amino acid residues. This has 4 amino acid or approximately 5 amino acid residues. This will form a fold, and this fold is shown here in schematic, and this fold is a beta sheeted fold, and this particular geometrically ordered unit is a secondary structure. Similarly, a set of amino acids here undergoes another folding, and this is also beta sheeted. So beta 1, beta 2, beta 3, beta 4, beta 5, and then this part is an alpha helix, so alpha 1. So this is how you read the secondary structure from the schematic, and each geometrically ordered unit consists of approximately 3 to 5 amino acids here. This is a particular protein sequence from E. coli, a bacteria, and you can see here they range between 3 to 5 amino acids, and each segment of 3 to 5 amino acids undergoes geometrical undergoes a particular folding in geometrical to form geometrical ordered units, and these units are called the secondary structure. And this particular figure here shows the secondary structure in much more detail. So it tells you this is the beta sheeted part, this is the alpha helical part, this is another alpha helical part, and this is the beta sheeted part. So by looking at the schematic, you can read the secondary structure of an amino acid, of a particular protein. So there are two types of secondary structure, alpha helix and beta sheet, and we will talk about both of these in detail. But what is the bond that is involved in the formation of secondary structure? The answer to this is the hydrogen bond. So primary structure, peptide bond; secondary structure, hydrogen bond. What are the types of secondary structure? Beta pleated sheet and alpha helix. You should be able to read the schematic of a particular protein which tells you the secondary structure, like the one I just showed you in this particular part in the slide. And if somebody is curious, what are these other lines? So what the person has done here is that he has aligned the same protein from different bacteria. So this is E. coli, this is Vibrio cholerae, the bacteria that is responsible for causing cholera. So, and you can see here there are minor variations in the sequence of the same protein across different bacteria. For example, here you have an I, which is isoleucine, but here you have a valine. But from this you can say that this is a conservative substitution, because both isoleucine and valine are non-polar amino acids. So this is why I am stressing on this, that you should be able to relate primary structure to secondary structure, and you should be able to appreciate that there are variations observed in the primary structure of proteins in the same species as well as across species. Coming back, secondary structure, folding of short continuous segments of polypeptides into geometrically ordered units. Two types, alpha helix and beta pleated sheet. The bond that is involved in the formation of secondary structure is hydrogen bond. If somebody asks you which of the following bonds is involved in the formation of secondary structure, peptide bond, hydrogen bond, electrostatic interaction, van der Waals interaction, the answer is hydrogen bond. This, you should not make mistakes on questions like this, because these are easy knowledge-based questions. So let us quickly look at the different types of secondary structure. We start with the alpha helix. So 3 to 30. So the stability of an alpha helix arises primarily from the hydrogen bond, and this hydrogen bond is formed between the oxygen of the peptide bond carbonyl and the hydrogen of the nitrogen on the fourth residue. So if I take 1, 2, 3, 4, you will see here that this is the nitrogen of the fourth residue, and this is the hydrogen bond that is formed in this particular alpha helical structure. And what is happening is that as we form the hydrogen bond, so these are hydrogen bonds that are formed. So this is a segment of a long helix. So these are the hydrogen bonds that are formed inside this alpha helix. And as this hydrogen bonding is taking place, a segment of the primary structure is forming the helix. And in forming the helix, the side chains are always facing out. The side chains are always facing out. And that's the reason here it is written side chains of amino acids in an alpha helix always extend outward. And again, you can see here why we stress on side chains of amino acids, because these side chains are responsible for assigning function to a particular secondary structure, in this case, the alpha helix. Now, the second most important thing that you should remember is that we talked about an amino acid proline, which is an amino acid which has a particular ring that you should remember. And I'm not going to reiterate what ring proline has, but proline is not an amino acid, it's an amino acid because it lacks hydrogen, and as a result it is unable to form a hydrogen bond unlike other amino acids. So what happens is, if in the primary structure there is proline, the primary structure will break. So which of the following amino acids disrupts secondary structure? The answer is proline, because proline is an amino acid. And this concept, again, we have revisited over and over again when we talked about amino acids. But this concept has to be translated now to the secondary structure, that because proline is an amino acid, presence of proline disrupts secondary structure, disrupts alpha helix, disrupts beta sheet. So if I do not see a secondary structure in a certain segment of the primary structure, and I ask you which amino acid do you think will be present in this particular segment, the answer is again proline. Depending on the direction of the helix, there are two types of helix: left-handed alpha helix and right-handed alpha helix. I will talk about this more in the in-class sessions, but if the left-handed alpha helix and the right-handed alpha helix, you have to just use your right and left hand, and the direction of the thumb points to the direction of the helix. And the fingers, when you coil your palm, the fingers point towards the direction of the coil. This we will talk about in the in-class session in a bit more detail. But depending on the direction, you have left-handed helix and a right-handed helix. There is a particular type of alpha helix that is not covered in your textbook but is often asked. It is called an amphipathic alpha helix. Amphipathic, amphibian, amphibious means having both. So amphibian animals means one animal that can live both on land and water. Amphipathic alpha helix contains both polar and non-polar amino acids. So in an amphipathic alpha helix, hydrophobic side chains project from one side of the axis of the helix, whereas hydrophilic side chains from the other side. So let us look at, so this is an alpha helix. This is a part of the helix. So if you look at this particular drawing, I am looking at the helix from top, not from the sides, from the top. And if you look at the distribution of amino acids here, these amino acids are predominantly hydrophilic whereas these amino acids are predominantly hydrophobic. So polar amino acids on one side, or hydrophilic amino acids on one side, hydrophobic amino acids on the other side. So this kind of alpha helix is called an amphipathic alpha helix. Sorry. So what I need you to do is refer to your book, Lippincott's Illustrated Reviews Biochemistry. That's your textbook. And read these two sections: amino acids per turn in an alpha helix and amino acids that disrupt an alpha helix. I mentioned that proline disrupts alpha helix over and over again, but please read this particular section in a bit more detail. You will be able to understand why proline is disrupting an alpha helix. Also, you need to know what are the number of amino acid present per turn in an alpha helix. The answer for this is 3.6, but please read these particular sections, and if you have some doubt, please write to me and we will go over this again. We come to the next type of secondary structure, the beta sheet. In beta sheet conformation, all the components of the peptide bond are involved in hydrogen bonding. You can see here, it's not like alpha helix where the first residue is hydrogen bonded to the third or fourth residue in the alpha helix. Here, each amino acid residue is forming a hydrogen bond. You can see here hydrogen bond, hydrogen bond. So in beta sheet, so what is the difference between beta sheet and alpha helix? In beta sheet, all components of the peptide bond are involved in hydrogen bonding. Whereas in alpha helix, it happens between specific specific amino acid residues depending on the position in the alpha helix. Now, beta sheet, or sometimes they are referred to as beta pleated sheet, because when we look at them in 3D, they look like pleats. So we sometimes people refer to them as beta pleated sheet, are of two types. We have parallel and anti-parallel. So in case of anti-parallel, you require two polypeptide chains. So this is one polypeptide chain. This is another polypeptide chain. The amino terminal of one polypeptide chain faces the C terminal or carboxy terminal of the other polypeptide chain. So you can see here, this is the amino terminal facing the carboxyl terminal, and this is polypeptide 1 and this is polypeptide 2. And therefore, since the amino terminal and the carboxyl terminal are in opposite sides, we call them anti-parallel beta pleated sheet, where the hydrogen bond is formed between the two polypeptide chains. Whereas in case of parallel beta pleated sheet, the beta sheet is formed, the hydrogen bonding is formed between the hydrogen bonding is formed within the same polypeptide chain. So you can see here, this is only one polypeptide chain. This is the amino terminal, this is the carboxyl terminal, and these are the short segments which are folded, and these foldings are interacting with the other fold by hydrogen bond, which gives it a sheet-like structure. And therefore, and the sheet-like, and since it is within the same polypeptide chain, we call it the parallel beta pleated sheet. Whereas in anti-parallel, there will be two polypeptide chains where the C terminal and the N terminal will be facing each other, and this kind of hydrogen bonding leads to the formation of a secondary structure which is called anti-parallel beta-pleated sheet. Take home message: anti-parallel requires 2 polypeptide chains. Parallel, you require only 1 polypeptide chain. Now, there is another type of secondary structure which is called a beta turn. In beta turn, what happens is the first participating residue, oxygen, from the carbonyl group. So this is C alpha. Remember, we talked about alpha carbon. This is the oxygen from the carbonyl group. And this is the amide linkage that is formed between 2 amino acids. So this is another amino acid, C alpha. This is the first residue. And this is the oxygen of the first carbonyl group here. And this will form a hydrogen bond with the 4th residue. So if I count it, 1, 2, 3, and 4. Each C alpha is 1 amino acid. So this is 1 amino acid, this is 1 amino acid, this is the 3rd amino acid, this is the 4th amino acid. I go here 1, 2, 3, and 4. I label my amino acids first, and then I look at the hydrogen bonding pattern. The hydrogen bonding is taking place between the oxygen from the carbonyl of the first residue with the 4th residue. And you can see here, this particular geometry that I get here is a turn. So therefore, you have a beta sheet coming and forming a turn, and this particular turn structure is called a beta turn. So the first amino acid, oxygen from the carbonyl group, hydrogen bonding with the 4th amino acid. And what kind of amino acids will you generally see in this turn? The answer to this are 2 amino acids are mostly seen in the turn. One is proline, the other one is glycine. So proline and glycine are 2 amino acids that are present in the beta turn. Very important. And why they are observed in the beta turn? Because proline, being an amino acid, cannot form hydrogen bonding. So if I want to have a proline in a sequence, I will have it either as a 2nd residue here or as a 3rd residue in the beta turn. Of course, not the 1st or the 4th residue. That you should remember. So amino acid, proline, and glycine are predominantly present in the beta turn. So this is a simple assignment. Which of the following interactions will promote the formation of secondary structure? I have been reiterating this over and over again. Disulfide bond, no. This is a bond that requires 2 cysteine thiol groups. This is not the one. Ionic bond, no. Hydrophobic interaction, also no. Van der Waals interaction, also no. The answer is hydrogen bond. And this kind of questions are always asked in an exam. So which is the predominant bond that is required for the formation of primary structure? Answer is peptide bond. Which of the following interactions will promote the formation of secondary structure in a polypeptide chain or a protein? The answer is hydrogen bond. Please, please, please do not make these mistakes in the exam, because these are like giveaway questions, and just by remembering specifics on the different level of protein structure, you should be able to answer these questions. We come to the last part of the lecture, which is supersecondary structure, or which is referred to as a motif. Now, what is a motif? Let us try to define a motif. A motif is simply a recognizable folding pattern involving two or more elements of secondary structure and the connections between them. I repeat, a motif is simply a recognizable folding pattern involving two or more elements of secondary structure and the connections between them. So let us look at the different types of motif that you commonly see in proteins. You have a helix, and then you have a loop, and then you have a helix again. So this motif is called a helix loop helix motif. You have a beta sheet, you have an alpha helix, you have a beta sheet, and this is called beta alpha beta motif. You have a beta pleated sheet, and then you have a beta turn, you have a beta sheet again, and these are anti-parallel beta sheets. Similarly, these are anti-parallel beta sheets connected by a beta turn, anti-parallel beta sheets connected by a beta turn. So a lot of beta, and therefore this particular motif, if you observe in a protein, is called a beta meander. You have parallel beta sheets, and these parallel beta sheets are forming a bowl-like structure, and this bowl-like structure that is formed is called a beta barrel. So why are motifs important? Why are we teaching you this particular part? Supersecondary structure or motif is important because it helps you to recognize a particular family of protein. It's like a signature. So if I see a particular motif, I can say, okay, this particular protein belongs to this family. And this will become more clear as we go into the higher orders of protein structure in the next lecture, that is tertiary structure and quaternary structure. But what I want to do here is to deconstruct a protein and show you what is the importance of supersecondary structure. So this is a protein, a channel protein. That means it's a transmembrane protein present in E. coli, and this particular protein is called tall C. Doesn't matter what it is called, but this is the structure of the complete protein that is shown here. And there is a particular database, you can go and explore this, which is called PDB, Protein Data Bank or Protein Database, where they have the structure of proteins. So what I'm going to do in the following slides is to deconstruct this particular protein. So if I start deconstructing, means I am just, let us take a ball of wool. Think about it as a ball of wool, and I am slowly unwinding the ball. And you can see here, as I deconstruct the protein piece by piece, bit by bit, I come to two basic supersecondary structures. Alpha helix. So we go back again, we have helix loop helix motif. So if I go and deconstruct this protein, you have a helix, you have a loop here, you have a helix here. So that's a motif that is present in tall C. And then if I go back again and I want to look at what other motif that I have learned is present in this protein, then I will find that I have another motif, which is beta alpha beta motif. Now if I see these two motifs after deconstructing a protein, I can tell that that protein is a channel protein, because channel proteins have this signature supersecondary structure. So in this case, this is tall C from E. coli, which is a channel protein. In fact, it's a channel protein. But any channel protein belonging to this family will also have these signature structures. They may have different primary structure, but they will have more or less similar secondary structure. That means they will have a helix loop helix motif and a beta alpha beta motif. So I go back again and I deconstruct it. So this is the tall C protein, and I deconstruct, and you can see here these are the two basic motifs that are present in this particular tall C protein, which is a channel protein. So any channel protein belonging to the tall C family will have these two motifs. So these two motifs become a signature for that particular group of channel proteins. So super secondary structures help you to identify the family of a particular protein. As I told you, it will become more clear as I go into tertiary structure and quaternary structure in our next lecture. So what I want you to do now is to read the section on super secondary structure or motifs from your book. And you can see here, the author of this book has written here, proteins that bind to DNA contain a limited number of motifs. So there will be certain motifs that will be present in these particular proteins. And the motif that is present in these proteins is the helix loop helix motif. So if I deconstruct a protein and I find that it is containing predominantly helix loop helix motif only, I can say that this particular protein most likely binds to DNA. So that brings us to the end of this particular lecture session. In our next lecture, we will talk about tertiary structure, quaternary structure, and we will also get an insight into protein folding. As I told you, primary structure and secondary structure are the initial structures when a protein starts to fold. So the primary structure is just the sequence. The bond involved is the peptide bond. A bit of folding of 3 to 30 amino acids into geometrically ordered units, and you get the secondary structure. The bond involved is hydrogen bond, two types, alpha helix and beta sheet. Alpha helix can be left-handed or right-handed. And then we talked about a specific helix called amphipathic helix, where you have polar amino acids on one side of the axis and non-polar amino acids on the other side of the axis. And what happens in an alpha helix is that the side chains are always extending outward. When we go to beta sheet, we have all the peptide bonds involved in hydrogen bond formation. And there are two types, anti-parallel and parallel. Anti-parallel, you require two polypeptide chains, N and C terminal facing each other, whereas parallel, you require a single polypeptide chain. We talked about beta turn, and we talked about amino acids that are predominantly present in the beta turn, that is proline and glycine. Which is the amino acid which disrupts secondary structure? Predominantly alpha helix. The answer is proline, because proline is an amino acid. And at the end of the lecture, we looked at super secondary structure or motifs which help us to identify a definite family of proteins. We have looked at helix loop helix motif, which is present predominantly in proteins that bind to DNA, whereas channel proteins like tall C have predominantly helix loop helix and the beta alpha beta motif. With that, I will end this lecture. Thank you very much for listening, and the other structures will be covered in the next lecture session. Thanks.